Since we opened it last week, we have our first sponsor, which is Horiba Scientific. So I really want to thank Horiba Scientific to believe in our concept of open science and want to support our activities. Thank you so much, Horiba. So with that, let's start on our sessions, and I'll pass it on to Xiaomei to introduce Zhijian.
Hello, good afternoon, everyone. We're glad to introduce today's speaker, Professor Zhijian Chai from Zhejiang University School of Medicine. So Professor Chai obtained his PhD degree in 2008 from the Zhejiang University School of Medicine. And then after 2 years of postdoc, he became a lecturer and then associate professor and was promoted to full professor in 2018. So also in the Zhejiang University School of Medicine. So now he's a full professor in the Institute of Immunology. So his research fields include tumor immunity, molecular immunity, regulation of immunity, autoimmune disease, and pathogenesis. In particular, he has been engaged in the studies of extracellular vesicles and immune regulations for a long time. So today, we're glad to have the opportunity to having Professor Chai here to give us this presentation. Welcome, Professor Chai.
Please go ahead.
Okay. Thank you for Xiaomei. The work I want to share today is extracellular vesicles, natural and safe drug delivery vesicles for liver diseases. As we all know, EV can derive from direct budding from the membrane, termed as micro-EV. EV can also derive from the endosome pathway and released as exosome. Because EV have a lot of proteins, information, genetic information, lipids, and metabolites, EV is very important in the information transfer between different cells. This article, maybe everyone is very familiar to this article. It's published by the Lottevo, we all know him, in 2007. He revealed for the first time that exosome can mediate the RNA transfer between different cells. And up to now, the citation is about 7,000. It's very amazing. It can say he opened a new research field for us. And this article is published by REC in 2008. In this article, he described for the first time that EV can mediate the protein transfer between different cells. And the citation is also very amazing. Based on these two findings, scientists start to use EV as a vehicle for drug. And in this article, which is published in 2011, would use siRNA-loaded EV to treat brain disease. After that, a lot of attention is paid on EV as the drug delivery vehicle for cancer. EV loaded with KRAS, KRAS mutation shRNA can be used to treat pancreatic cancer. And the EV loaded with chemotherapeutic drug or EV loaded with anti-microRNA also show excellent anti-tumor effect. Use EV as drug delivery vehicles has a very good perspective because EV comes from the cell. So EV are less immunogenicity and lower toxicity. EV have higher biocompatibility and higher engineering potential. Namely, we can modify EV to make the EV have the ability to targeting specific disease, or we can load specific drug into the EV for the related disease. EV can also cross various biological barriers, including blood-brain barriers. EV can also escape from lysosome degradation. However, if we use EV as the drug delivery vehicles, we also face a lot of challenges, including the source of EV. Which kind of cell we select to generate EV is important. And the isolation of EV, because up to now, no one can isolate EV with 1% purity. So to include the EV purity is also very important. We can load a lot of drug, include siRNA and chemotherapy drug into the EV. But how to load these drugs with high efficacy is very important and needed to be solved. If we use EV to treat specific disease, the EV need to have the ability to target to the disease. So the tissue specificity of EV is also required. In addition, for clinical use, the biological safety and the clinical grade production of EV is also required. To increase the possibility of EV as drug delivery vehicle in clinical, two main problems need to be solved. The first one is the biological safety of EV. And the second one is the targeting of EV for disease. A lot of publication have reported that EV can be artificially modified to targeting tumor. For example, tumor cell express very high level of alpha-5 integrin. So alpha-5 integrin specific IRGD peptide modified EV can specific target to tumor and show powerful anti-tumor effects. AAPG is the ligand of the lung cancer cell sigma receptor. So AAPG modified EV can specific target to the lung cancer and show better anti-lung cancer effects. Like CAR-T, EV can also be modified by HCFV and can be target specific targets to the tumor antigen specific expressing cells. As for the biological safety of EV, RBC-EV is an ideal candidate. This is a publication published in Nature Communication. In this article, it mentioned that RBC-EV lack both lack DNA, so they can avoid the risk of gene mutation. RBC are the most abundant cell type in the body, so we can conveniently obtain large-scale EV production. RBC-EV are the components of blood, and blood transfusion are of high safety. So the RBC-EV is also very safe. Given the excellent property of RBC-EV as drug delivery vehicle, so we further investigate the in vivo distribution of RBC-EV. First, we isolated RBC-EV and identified the EV from the protein contents, morphology, and the size distribution. We confirmed that the RBC-EV we obtained were exon-like vesicles. Then, after the vivo track labeled RBC-EV is intravenous injected into B6 mice, we found that the EV is natural liver accumulated, and this phenomenon is more obvious over time. We also confirmed this phenomenon by tissue section when viewed by microscopy. Generally, the blood transfusion is allogeneic. So this is about we concentrated the EV by sucrose gradient and found the concentrated EV were also liver accumulated. However, the vivo track alone is non-specific distribution in vivo. The liver is the organ that's rich in bloodstream. So the injection we select is intravenous. So to exclude that the anatomical structure will infect the accumulation of RBC-EV. We also IT or IP injection RBC-EV into B6 mice and found by these two ways RBC-EV could also accumulated in the liver. Generally, the blood transfusion is allogeneic, so we transfer RBC-EV from Barbie mice into B6 mice, and we found the allogenic RBC-EV were also liver accumulated. Even more, when human RBC-EV were transferred into B6 mice, they were also liver accumulated. Because the production of RBC-EV, we use the human RBC-EV for the following experiment. We also transfer EV from 41 breast cancer cell or EV from dendritic cells, we found both EV were liver accumulated. This result indicate that liver accumulation is the common property of EV, but not specific to RBC-EV. Then we want to know why the RBC-EV is liver accumulated. First, we want to know which cell subsites in liver uptake RBC-EV. We found the alpha SMA positive stellate cell and CD31 positive endothelial cell and the albumin positive hepatitis did not uptake EV. But the F480 macrophage greatly uptake RBC-EV. This was further confirmed by the flow cytometry. We can see the vivo tract positive liver cell is the macrophage. Then we depletion the macrophage by clodosome. We found after depletion of macrophage, the liver accumulation of RBC-EV is decreased, along with the increase of RBC-EV in other organs. Next, we want to know why this will happen. We assume that the liver environment will affect the uptake of RBC-EV by macrophage. So we couch the macrophage with lung, liver, and kidney lysates. As expected, we found that the liver lysates significantly promoted the the macrophage to uptake RBC-EV. And the lysates from liver cancer cell line Hep1/6 can also promote the macrophage to uptake RBC-EV. And the complement is synthesized in the liver, and the CEQ is one of the complements. And the CEQ is considered as one of the immune signal. We confirmed that CEQ in the liver is richest. Then by CEQ inhibit DHP, we found DHP can abolish the effect of lysate of Hep1/6 to promote macrophage to uptake RBC-EV. So did CEQ siRNA. More directly, CEQ treatment can promote the macrophage to uptake RBC-EV. So CEQ in the liver promotes macrophage to uptake RBC-EV in vitro. Then we also investigate the role of CEQ in vitro. We found that when the CEQ in the liver is inhibited by DHP, the liver accumulation of RBC-EV is also greatly inhibited. And the macrophage that uptake RBC-EV is also decreased. Moreover, we found that the numbers of macrophage in the liver is the most. So the liver environment and the number priority of macrophage in the liver both contribute to the uptake of RBC-EV. To extend our finding to human, we co-culture the human liver, lung, and kidney tissue with the WebEVTalk labeled EV. We found that liver tissue have the strongest ability to uptake RBC-EV in vitro. And the main cell that uptake RBC-EV is also human macrophage. We also confirmed that the human liver lysates have the strongest ability to promote macrophage to uptake RBC-EV. Similarly, this effect could also be abrogated by DHP. And the human CQ could also promote the macrophage to uptake RBC-EV. Moreover, the macrophage number in human liver is also the highest. MicroRNA 155 can promote the inflammatory injury in the liver. So we assume that anti-155 loaded RBC-EV probably can be used to prevent acute liver failure. So we loaded anti-155 into RBC-EV and pre-transfer then into the mouse and induce the acute liver failure. We found anti-155 loaded RBC-EV greatly inhibits the microRNA 155 levels in livers of ALF mice. And in the histopathology results, we can find the anti-155 loaded RBC-EV show greatly preventive effects to the liver damage. And the liver function and the pro-inflammatory cytokines results also support this result, support this notion. Moreover, we found that the anti-155 loaded RBC-EV can promote the N2 polarization and inhibited the N1 polarization, which is beneficial to the inhibition of inflammation. Then we want to know the anti-155 loaded RBC-EV can use to treat the acute liver failure. So we induce the acute liver failure first, and 6 hours after the induce of acute liver failure, we transfer the anti-155 loaded EV and found that it show obviously therapeutic effects to the ALF. So anti-155 loaded RBC-EV can be used to treat acute liver failure. Because macrophage is responsible for the uptake of RBC-EV in the liver, so we want to test the role of macrophage in this process. We deplete macrophage and found after the macrophage depletion, anti-155 loaded RBC-EV could still downregulated the microRNA 155 level in the liver. However, the effect was great inhibit, which can also reflected by the histopathology results and the liver function and the pro-inflammatory cytokine results. So the therapeutic effect of anti-155 loaded RBC-EV on acute liver failure is mainly macrophage dependent. Then we want to know if the RBC-EV loaded with anti-tumor drugs, they probably can be used to treat orthotopic liver cancer. So we loaded doxorubicin into RBC-EV and confirmed that the doxorubicin in RBC-EV is stable at least for 3 months when resolved at minus 80 centigrade. Then we confirmed that the RBC dox could greatly inhibit the growth of liver cancer cell in vitro, and this effect is comparable to the loading dose dox. Even if the concentration of the loading dose dox is 10-fold greater than the dox in RBC-EV. Then we transferred the RBC-EV dox into mice with orthotopic liver cancer. We found that the RBC-EV dox could greatly inhibit the growth of liver cancer, and this effect is better than the loading dose dox. And the dose of loading dose dox is 30-fold greater than that in the RBC-EV. However, the RBC-EV dox exhibits better therapeutic effect. Then we depleted macrophage and found the therapeutic effect of RBC-EV dox is abolished. So the therapeutic effect of RBC-EV dox is macrophage dependent. Sorafenib is the first line drug for liver cancer. It can inhibit the growth of tumor and inhibits angiogenesis. So we also loaded sorafenib into RBC-EV, but we only found the slight inhibitory effects in the growth of liver cancer cell in vitro by RBC-EV sorafenib. However, the RBC-EV sorafenib can greatly inhibit the angiogenesis in vitro. Similar to RBC-EV dox, the RBC-EV sorafenib greatly inhibits the growth of liver cancer, and this effect is better than the loading dose sorafenib. Moreover, RBC-EV sorafenib greatly inhibits angiogenesis in tumor tissue. So anti-tumor drug-loaded RBC-EV can be used to treat orthotopic liver cancer. Finally, we want to evaluate the biological safety of the drug-loaded RBC-EV. The intravenous injection of anti-155 RBC-EV did not cause obviously histopathology damage to the main organs, including the heart, liver, spleen, lung, and kidneys. Interesting, even when accumulated in the liver, anti-155 RBC-EV did not increase the ALT and the AST, but inhibited them. For RBC-EV dogs, we found that weight loss and weakness were only observed in mice that received the RD-dox injection. The main side effect of dogs is cardiotoxicity. We can see RBC-EV dogs did not significantly increase the level of CKMB and the LDH. However, the RD-dox led to significantly cardiac damage. Compared with the RD-dox, RBC-EV caused more severe impairment of liver function. This suggests that the improved accumulation of dogs in the liver. But from the results of histopathological, we cannot see obviously damage of the liver tissue by RBC-EV dogs. But we can see obviously the damage of the heart tissue by RD dogs. Sorafenib had been reported to cause severe skin toxicity. We found that the skin from the RD sorafenib treated mice showed increased thickness and downward extension of the stratum spinosum. But neither the RBC-EV sorafenib and nor the RD sorafenib affected the angiogenesis in the heart, liver, spleen, lung, and the kidney. So this result suggests that the drug-loaded RBC-EV have a very ideal biological safety. Then we concluded that RBC-EV are natural liver accumulating. RBC-EV loaded with anti-155 can celebrate acute liver failing. RBC-EV loaded with dogs or sorafenib inhibits orthotopic liver cancer growth. RBC-EV are natural delivery vehicles of therapeutic drugs for liver diseases. We also use RBC-EV as the delivery vehicles for the treatment of liver fibrous. Last, I want to thank Dr. Gensen and Xiaofang and Hui Qing for their work to this article. And I want to thank everyone for hearing my share.
Thank you very much, Professor Chai, for this very informative lecture. Now is the time for question. We have, we have Isaac Shep, his question. I don't know how, can you hear? The question is regarding
Isaac Cai, can you unmute yourself and ask your question?
I don't know how to switch to his screen. Carolina, can you see you can see the question, right?
Yeah.
Yeah.
I asked Isaac to unmute, but she's not. Okay. Should I read the question?
Maybe just we read the question.
Yeah.
So Professor Chai, there is a question regarding false positive signal of lipophilic dyes, such as PKH or DIR, and the low signal intensity of other labels. What do you suggest for EV labeling to get a reliable signal for biodistribution studies?
Okay, you mentioned the signal in other organ is very low.
Which means how do you use fluorescent label to distinguish extracellular vesicles from the background, so you can see the distribution? Yeah. But what the question is for biodistribution studies.
Okay. I use PKH 26 to label the EV for the microscopic detection. How to label the EV? I don't know that detail.
Do you mean if you use PKH, which will give you good enough sensitivity under the microscope so that you will be able to see the EVs, the individual EVs?
Yes, I use the label, the dying of PKH 26 to label the EV, but I don't know the detail how to label it.
The students do the experiments.
Yes.
Yeah, okay. We have another question, Dr. Lee. So the question is, how do RBC-EVs escape the powerful lysosome system in macrophages to deliver the microRNA 155 ASO to the cytoplasm? And how is DOX transferred from macrophage to other cells?
Okay.
Yeah.
Thank you for reading the question. It's nice to talk. I'm very glad that you have done a lot of work to illustrate the system. I think it's very reproducible and also in our lab that this red blood cell extracellular vesicles go to macrophage. They are taken up a lot by macrophage, especially in the liver. But we have not seen very good effect in the macrophage when we deliver RNA, because I think it's very difficult for RNA to escape from EVs. without being degraded in the lysosome, in the macrophage, because they have very powerful lysosomal system. So I wonder how you have you done any experiment to check the escape of the ASO into the cytoplasm, and how does this work? Also, when you talk about doxycycline and sorafenib, if you show that the effect was mediated by macrophage, then how do the drug kill the tumor cells? How do the drug transfer from macrophage to tumor cells?
It's a good question. We did not do the experiment to detect how the microRNA in the EV escape from the lysosome, but the anti-155 loaded RBC-EV can inhibit the micro 155 levels in macrophage in vitro and in vivo with very high efficacy. So I think it's not difficult to repeat this experiment. And for the doxine and sorafenib, why they take up, they are uptake by the macrophage, but can injure the cancer cell. We also do some. We had to do some experiments to explain this phenomenon, and we stain the macrophage and the cancer cell after injection. After injection, the drug-loaded RBC-EV. We found the drug before 24 hours before the after 24 hours after the drug injection, the drug mainly is located in the macrophage, and during this time, the apoptosis is occurred in the macrophage, and after 24 hours, the drug is found in the liver cancer cell, and the apoptosis then it occurred in the liver cancer cell. And in one publication, which is published in Nature Communication, in this article, it's claimed that when the cell uptake the drug-loaded EV, they can also secrete EV with drug. This is the second generation of the EV. And for the third generation of EV, they can also carrying the drug. So this just like a taurine effect.
Oh, that's cool. Have you tried to elucidate that if the drug that go to macrophage, and then if you harvest the EVs from the macrophage, do you find the level of the drug in those EVs higher than in the control?
Yes, we found the drug-loaded EV in the macrophage. The signal of the drug-loaded EV in the macrophage is higher than the control treatment.
Wow, okay, that's great. Thank you very much.
Thank you.
Yeah, yeah, we have some other questions from Masha Sale. Can you? So the question is, I want to know, did you measure the hemolysis percentage during the RBC-EVs isolation? You can. Yeah, hemolysis.
Hemolysis as percentage.
溶血，血细胞溶解。
Oh.
嗯
We did not do this experiment, and when the EV is isolated and transferred into the in vivo, we did not find some abnormal from the mice. So we cannot. I cannot answer your question, but I think this likely is very low. This possibility is very low, I think.
We have one more question. What is the efficiency of microRNA loading, and how to separate the free microRNA from the loading mixture?
The efficiency of the siRNA loading is about 10 to 20%. And we use the ultra centrifugation to isolate the free siRNA from the loaded siRNA.
So you use 100,000 G with 2 hours centrifugation?
Yes.
You find aggregate after that? That is a very long centrifugation.
We use 100,000
gram, yeah.
gram to centrifugate for 1 hour.
Only 1 hour?
Yes, only 1 hour is enough.
Okay. And one more question is how the ASOs were loaded, and did you know any aggregation of RBC-EVs?
The NT155 is loaded by electroporation. We did not see the aggregation of the RBC-EV during this process, but we did see the aggregation when we loaded the doxorubation into the RBC-EV.
Okay, yeah, I have one more question. For you described different kinds of drugs, and what would be the most efficient loading strategies for each kind of them?
Yes, I think the best loading protocol we used is electroporation. We also, but I think it's not good enough. I think this way is not very stable. So we still explore one way to stable and efficient to load the drug into the EV. We have some exciting finding. We will publish it soon.
Great. We look forward to your new publication.
Thank you.
Okay. If there is no other questions, let's thank Professor Chai for his wonderful presentation, and also thank all the audience for your participation. And thank Carolina for the organization of this wonderful series EV-WebEVTalk events.
Yeah, thank you so much, Professor Chai, for sharing your result with us and your experience on working with the EVs for liver disease. And also Xiaomei, Yan, Professor Xiaomei Yan, thank you so much for leading the sessions today and leading the question and answer.